Our second speaker is Dr. Shirin Doroudgar. She is a PhD at University of Arizona. She works as an associate professor in the University of Arizona College of Medicine, Phoenix's Department of Department of Internal Medicine, and is an active member of the Translational Cardiovascular Research Center, where she leads a research group in cardiac molecular biology, focused on understanding changes in protein homeostasis and cellular stress responses that contribute to heart disease. So welcome, Dr. Doroudgar, for your presentation.
Thank you so much for that kind introduction. I'm very happy to be here today. Thank you to Dr. Dev and organizing committee for having me. I'm excited to talk to you about a topic that I'm very passionate about and I work on in my lab. And I'm very fortunate that my mentor who got me started in this field, Dr. Glombotsky, is here today. So we started working on protein homeostasis in the heart a while ago, and I'll be debating him later this afternoon, so don't go anywhere. But we'll open with some of the some topics that might set us up for that. So how and when to measure TTRs. I have no financial disclosures. So we've seen this diagram a number of times. We've learned a lot today. In addition to what we've known before about this process. So the majority of the TTR that's circulating in the blood comes from the liver, as we know, and that exists in a healthy individual in a folded or native state. And as we all know, that folded native state is susceptible to falling apart into these monomers, which then can misfold and in different tissues can aggregate. And what I'd like to draw your attention to is, of course, we know the stabilizers are blocking this dissociation, but the disease process and what's important is understanding what's going on here. So understanding, earlier disease processes and how these soluble precursors, these monomers, can deposit as insoluble fibrils is very important. And so these fibrils are seeding the addition of other fibrils, and this can continue with age, as we know. So there is a gap that we call the monitoring gap. So that's how we monitor ATTR cardiomyopathy care, but there's a gap there. So we would like to stratify patients with ATTR cardiomyopathy. We know this is critical for optimal management and treatment. However, monitoring disease progression is challenging and currently lacks best practice guidance in some ways, at least in relation to the TTR measurements that we're going to talk about today. So what I'd like you to think about is that we would like to consider TTR regulation across multiple levels of protein homeostasis. I study protein homeostasis in my lab, not only in therapy development, but also in monitoring disease progression. So the protein TTR is, synthesized, it's folded, it's secreted, and it can be degraded, and its quality is controlled, and its levels can be affected by changes in all of these different levels of regulation. So what can be measured? We've seen earlier in this tiny diagram, these are biopsies that can be tiny punch biopsies that can be looked at with a stained Congo red, which is not specific for TTR, but there are also TTR antibodies that can be used on the same kind of biopsies. And here they're stained in brown, indicating the presence of this TTR amyloid. So this is not a very quantitative, sensitive way to measure TTR. And of course, it's not always feasible, and we can't do this over time to the same patient. So I'll talk to you about these 4 different ways of measuring TTR protein, which is different than using these punch biopsies and immunohistochemistry like these brown stains. So first is total circulating TTR concentrations and stability, and we'll go over the methods that are mostly used in the clinical setting. In the research area, we have some methods that are developed to look at TTR tetramer stability, but they're not really amenable to clinical care, so we'll see why. And then we can also measure misfolded or amyloidogenic species indirectly, and also genetic variants in protein can also be assessed at the protein level, not just at the DNA level. So total circulating TTR concentration and stability, the methods that are used right now, a lot of them are relying on immunoassays. So this is the most common in clinical practice. Some of the studies we've looked at earlier today rely on these turbidimetry or turbidimetric immunoassays. And this measures the decrease in intensity of light that's transmitted through a sample because the sample becomes more turbid. And that turbidity is caused by TTR that's present and a TTR antibody, and this happens in a test tube. So we set this up and we can measure the amount of TTR that's existing, for example, in the plasma that we get from a patient. As I said, this is widely used in labs and hospital labs, and it measures total circulating TTR. It's fast and standardized, and it's relatively inexpensive, and you might know it as pre-albumin measurements. There's a modification of this, which allows us to measure stabilized TTR. And so this is performed on platforms that we know of, such as Abbott Architect. So these systems are using a modified immunoturbidimetric assay. And this is a special tool to look at intact native tetrameric TTR in human plasma. But what we have to remember is in order to look at the stabilized forms of TTR, we're not just measuring the levels of it, we're looking at how much of it stays stable. We have to denature it. So we have to do something like adding urea or acid that denatures the protein. And that kind of is what gives us the, what stabilized in terms of TTR stabilization. So I want to just take us into now the most common TTR tetramer stability assays in research. What can we do in the lab? We don't do all of these things in the clinic, but these assess how resistant native TTR is to dissociation into monomers again. So looking at the earlier stages of dissociation, and you might be familiar with fluorescent probe exclusion. So this is a method we can use in the lab that it uses a competitive binding of a probe to TTR. And the probe is fluorescent when it's bound to TTR. Now, I have an example of the next one here. This is Western blotting. A lot of times when you're a basic scientist, you use Western blots to look at the quantities of proteins, but you can also look at dissociation of tetramers by doing a certain kind of a Western blot under denaturing conditions. And that's shown here in this Western blot figure where proteins are run on a gel and separated by size. And then here they're denatured. So this condition is denaturing, but you can see that tetramer going away, but with the stabilizers here, the tetramer is more stable. And this is an example of how Western blotting can be used, but we do this in the lab mostly in the experiments. What these conditions a lot of times rely on, so I mentioned denaturing conditions a couple of times in terms of stability assays. These actually perturb the ligand affinity as well as TTR stability. So they underestimate the stability of TTR. And so there's a gold standard called subunit exchange, which is drawn here on this diagram, where this is done in a test tube where we can engineer the TTR to have a tag and then add it to untagged monomers. So one is tagged, one is untagged, and this incubates, in our test tube for a while, and then we can measure the subunit exchange. And the less stable the situation is, the more of those subunits gets exchanged. So the good thing about this assay is that it's the only method that measures TTR kinetic stabilization under physiologic conditions and doesn't really rely on the kind of denaturations that might actually destabilize TTR as well. Okay, so next I'm just going to touch briefly on misfolded or amyloidogenic species and how we can measure those. These are done now mostly in the research setting again. So there's been studies that have been published that have come up with conformation-specific antibodies, and these can distinguish between folded or misfolded types of TTR. These are not used in conventional routine TTR diagnostic turbidimetric assays that we mentioned in the beginning. The problem with this is these TTR species, the pathogenic species that we're interested in, I showed you those earlier, misfolded monomers and oligomers, those are very transient, they're low abundant, and they're dynamic, so it's difficult to pick them up with these conformation-specific antibodies. And so recently, there have been a lot of investigations into identifying conformation-specific probes that individuals have developed, researchers have developed, that reveal more earlier on, more relevant TTR instability earlier than antibodies. And so lastly, what can be measured in terms of genetic variants? What's the most useful is looking at the protein sequence itself. And we can do that using liquid chromatography coupled to tandem mass spectrometry, also called LC-MS-MS. And this can quantify TTR with high specificity, and you can sequence, read every individual amino acid that makes up that protein, and you can distinguish wild type versus variant, and also various post-translational modifications. And this is mainly used in research or in very specialized centers, and it's not routine in clinical care. Okay, so when do we want to measure TTR? When we look at the American Heart Association, we are faced with this question that they're asking at the moment: are there biological processes in TTR stability, TTR kinetics or levels, or TTR ligands that can be used to monitor progression? It's posed as a question, it's not a guideline yet. And then when we look at the American College of Cardiology, they ask a similar question, and so they suggest that we would like to determine contributions of potential indicators of therapeutic efficacy or disease progression in multiple complementary domains, including circulating biomarkers, which also include TTR, troponin, and natriuretic peptides. So there's no clear guidance yet. The questions are out there. We know that in routine clinical care, we've already heard about trials this morning, that TTR measurement remains largely trial-based and is typically performed using that immunoturbidimetric assay, which is easy to do or quick to do compared to some of those other biochemical assays that we went through. And circulating TTR values may increase as systemic inflammation decreases or metabolic status improves. So that's something to definitely keep in mind when measuring TTR levels. And then in terms of interpreting TTR levels, there are determinants of circulating levels of TTR that definitely remain unexplored. I refer you to the beginning slides where there's all those different levels of protein regulation that can affect circulating TTR levels. Protein synthesis, quality control, secretion, Dr. Shirin mentioned this morning, degradation and how it deposits in different impacted tissues all affect circulating TTR levels. And what's ultimately very important is to understand how much of it deposits in an organ, and that is very difficult to measure over time right now. And so context is also important. Low TTR can be a marker of also malnutrition, inflammation, and metabolic issues, so those have to be taken into account when interpreting TTR levels. So in summary, there are multiple methods to measure TTR levels and stability, but most were developed for research rather than routine care, so they're a little bit more difficult to implement in the clinical setting. Few of the assays are fast, standardized, or clinically validated for longitudinal patient monitoring. The assays that rely on turbidity are currently the most standardized approach for measuring circulating TTR, but may not reflect the extent of TTR stabilization under physiological conditions. And in clinical practice, TTR measurement is largely trial-driven, and interpretation is limited to circulating protein levels. TTR stabilizers raise circulating TTR, as we've heard earlier, yet plasma levels may not reliably reflect tissue amyloid burden or clinical response. And effective disease monitoring requires integrated assessment. So we need biomarkers, imaging, and function And this highlights the need for evidence-based and guidelines and direct measures of amyloid burden. So I think with that, I will stop and take any questions that you might have. Thank you.
Any questions for Dr. Doroudgar?
So in a clinical setting, what types of measurement tools can be used to track the disease progression? And I remember a couple of years ago hearing about a doctor in Tennessee that had a specific type of imaging that showed amyloids deposited in different parts of the body, and that there were no other places other than maybe in Europe that did that. And I'm not sure if this is the right question for this discussion, but that putting it out there.
Maybe Dr. Doroudgar may have an answer here.
I can try. So we definitely, so imaging is great, and I don't know about the sensitivities of maybe the imaging that you're referring to. They may not reflect the short-term changes that we might want to follow up on, or earlier detection that we would like to have with before we start treatment. What we're interested now in is to identify additional biomarkers that might indicate in more sensitive ways cardiac damage earlier on. So that's kind of where we're going, or our research is going towards identifying specific biomarkers. The biomarkers that exist now, in addition to TTR, troponin, or BNP, those are not non-specific and may not be helpful for following up and for treatment response. So we're interested in understanding more about the biomarkers and how the cells respond, and maybe release signals that are specific to damage by these kinds of misfolded proteins, and maybe even specifically TTR. But now I'm going back to the basic research part of it, because that's where I see the greatest need for development right now.
So it was fantastic talk and very provocative topics that you covered. And I'm wondering, I have lots of questions for you, and I think there's a debate this afternoon. So it sounds like a lot of con points that you've made. And so I appreciate that, because I'm on the con team. But
I didn't mean to do that.
What I
put my
really
foot in my
wanted
mouth.
to ask you was more, you said this is research-based several times in your technologies. And we all know it usually starts there, but has to develop somehow into a more clinical lab application. And so one question was, I wondered what your realistic vision is for the future, and how many months or years is that vision going to be played out as to how we can really hone our analyses of the right biomarkers for TTR. The second thing is, we talk about TTR when it misbehaves, but when it behaves, it's a necessary component of physiology. So I wonder if when we undo the misbehaving or reverse it or whatever it is we're trying to do, how are we going to affect normal physiology? So that's kind of like two questions in one.
That's perfect. Maybe I'll answer the second one first. So when we fix the problem, so when we fold or stabilize TTR, I believe that we're improving, systemic physiology, and I touched upon it. There are, potential areas to investigate in terms of looking at metabolism, positive changes in that direction, and inflammation and things like that could happen that are part of that process of improving that protein's synthesis, folding, secretion, circulation, and its functions downstream. So that can have a whole system, benefit in ways that we may not even understand yet. In terms of, your first question about how things start in the lab, and the turbid assays started in the lab too, and went into the clinic, and they were now easier to do there and applied in that context. And this goes back to the first question as well. the assays that I would do, so if I could spend my time developing an assay, I would take the assays that we take too long to do in the lab on, if we were to apply to many samples. So for example, the subunit exchange, and apply it to our patients. So if if we're interested in TTR levels and stability in plasma and following that up after treatment, we can add the preclinical assays that we do now. So there's the subunit exchange, for example, the gold standard, maybe automated it a little bit more so that it's easier to do for the patient samples, and add it to the turbidimetric assays. And it might even give us a bigger difference. So we might even be underestimating how much TTR is stabilized when we do use the turbidimetric assays, which denature the protein and maybe actually negatively affect the assay.
Thank you so much.
Thank you.